Kumar University for providing me to give a small lecture as a short-term video for presenting some topic. Very good morning to all of you. Today we will study about the life cycle of parasitic plant. As we know that plants are two types, autotrophic and parasitic. Autotrophic plants do photosynthesis and they complete their life cycle, like after the germination plant grow and they give flower, and after flowering pollination followed by fruit formation and seed maturation. This seed set on the soil and it give new plants. But in case of the parasitic plant, they cannot do the photosynthesis. So these parasitic plant live on the other autotrophic plant for their life support. In that case, how a parasitic plant can identify the autotrophic plant, that some autotrophic plant are growing nearby, so that they can, after germination, they can directly attach to the autotrophic plant. Because generally the seeds of parasitic plants have very less amount of the endosperm, so they need to attach directly to the autotrophic plant just after their germination. So the parasitic plants adopt a strategy to identify autotrophic plant growing there nearby. As we can see, in case of Orobanche, it produce seeds, huge amount of the seed, and they can live in the soil about 20 years. These seeds detect the autotrophic plant by histocolactone. These histocolactones are produced by autotrophic plant in search of the endophytic or mycorrhizal fungi, and these Orobanche seeds use this potential of the histocolactone production by autotrophic plant to detect the autotrophic plant growing nearby. As after the imbibition of the seed germinate, and as it find the histocolactone, it means there is some autotrophic plant, and they directly attach to their root. If suppose that there is germination of Orobanche seeds and there is no histocolactone, then it can reverse and it can again convert themselves to the dormant state. So Orobanche or other parasitic plant develop a strategy to identify the autotrophic plant for their survival, and this is very interesting strategy which can be seen in our nature. But we can also use this strategy to produce development of resistant autotrophic plant against the parasitic plant. Suppose that we can develop a autotrophic plant having less histocolactone producing by their root, we can develop a technique which can be tolerable or resistant against these parasitic plants. Thank you.